Hello friends, this is the second part of the video where I will show you the experiment, the part 1 which was there related with the materials, and I have briefly explained about the principle. So in this part we will see that how we can perform the conjugation experiment. Different people have done different versions and it varies from lab protocols, one lab to one labs, they have different protocols. Second thing, the resistance or the selectable marker genes which people usually use also differ from one lab to other. In our case, we are using a donor strain which is a tetracycline resistant, and the recipient strain which is your streptomycin resistant but sensitive to tetracycline, and then it will transfer. So how we do this experiment? So for this, one has to do a overnight grown culture, and after growing overnight culture they have to inoculate into a secondary culture and allow the cells to be a log phase cells. So the rest of the things which I will be showing in the laminar hood, because we have already done this thing, we have already revived the strain, we have grown it overnight, and we have also given a 1 hour incubation that cells are in a log phase cells. Now how to perform the experiments, that's which you are going to watch, so please stay tuned here. So first of all, one has to sterilize his or her hand, and if you are wearing any kinds of rings or any watch, then it should be removed. Like as you can see, I'm trying to improve. I just put a pull over, then this is my sterilizer. Okay, so in this sterilizer I will properly see that my hand should be sterilized. Now the first thing which is required is your donor strain, which I already explained in my part 1. This is my recipient strain, this is my donor strain which has already been grown, okay, for log phase, and this is my recipient strain which is already grown at the log phase stage. Now this is my plates where I'm going to pour this thing, okay, and the second one is my streptomycin plate for as a control, and I have a 2 antibiotic strains with me over here, okay, as you can see, which is consist of both tetracycline and as well as my streptomycin antibiotics added. So the first thing which I'm going to do here is, so these are my Appendrof as per the protocol. I will label this as a donor. So let's say this is my number 1, this is number 2 is recipient, number 2. Then I have here autoclaved, because we routinely do it, so we have autoclaved, as you can see, the tip box, okay. So I will take 500 microliter of log phase donor strain. So this is my donor strain, okay, as you can see. So 500 microliter of my donor strain, okay, this I have taken. This is in the log phase cell stage, which is having a OD of 0.4 to 0.6. It has already been measured. This is my recipients, okay, so already grown for 1 hour at 37 degree. Again, this is the 500 microliter. You have seen that I have changed, okay, you have seen that I have changed my tips to avoid contamination, and since I am inside a laminar hood and there is a kind of the chances of contamination will be very less. Then what I am going to do here is, I will take another tube, okay, this is my tube, and this tube I will keep it for say mating, or you can say the conjugation. So this log phase donor strain, here I will take into this. So I am doing a liquid broth kind of a mating experiment, and I'm not changing the tip because actually I'm going to mix them. So this is my recipient strain, and I will add 500 microliter and 500 microliter, and this is done. So what I'm going to do here is I'm going to put this thing in 37 degrees Celsius for at least 20 minutes. Okay. Okay, friends, welcome back. So this is 20 minutes is over. I allowed the conjugation to happen. Okay, so it was in the water bath here, if you can see. Now so this is my tetracycline donor plate. Though this is just to make sure that everything is fine, there is no revertants which basically formed. So this is one plate. This is also my recipient streptomycin plate. So there should not be any revertant back. So that's why as a negative control I'm putting. And this is my original plate which I have to do it. Okay. So what I'm going to do here is, so now I will take, this is as my 100 microliter pipette. Okay. I will take my mating, this is approximately our conjugation which took place. 100 microliter of this I will plate in my tetracycline plate here in the middle, as you can see, and 100 microliter in my plate where I want that it should work. So this is my plate number 3, and actual experiment where it is taking place, this one I'm going to put. Okay. So then I will throw this. So as you can see, there are 3 plates. Okay. So these are my sterile, you can say, spreaders which we use it. So what I'm going to do here is that I will mix it. Okay. So I will mix this. I'm showing you like this. Okay. So I will spread plate it. So these are kind of a disposable one, so we can just throw them. This is the another one. And I will also spread plate this, where I expect that if the conjugation occurred, there should be something. Okay. And then I will also discard this, and the third one is my streptomycin plate. So this one, so this is I am also showing you in the top so that you can see it. Okay, you can tilt it, that's not a problem. So this will be mixed. So these are my 3 plates. Now these plates are ready to be gone into the 37 degrees Celsius, and we will see by tomorrow if the conjugation occurred, some kinds of results will be there. So this will go in 37 degrees. So in the middle plate where I have put both the antibiotic, if my conjugation experiments work fine, then I expect colonies. And how to calculate the conjugation efficiency, for that you can watch my another video. So with this I will stop. Thank you very much, and thanks for watching this video.